Welcome to the convening of the California Dyslexia Initiative Professional Learning Network. I'm Dave Gordon, the Sacramento County Superintendent of Schools. On behalf of the California Dyslexia Initiative, I want to welcome you to the 2024 Spring PLN. The goal of the CDI is to build capacity and resources across California's educational systems in order to address the needs of struggling readers and students at risk for and with dyslexia. The CDI is doing this through specialized professional learning opportunities, an annual webinar series featuring experts in their field, developing a repository of tools and resources, a conference this June, and what you are here for today, statewide professional learning networks. Today we have the honor of learning from Dr. Tim Odegard, Murphy Chair of Excellence and Professor of Psychology at the Middle Tennessee State University. Dr. Odegard's keynote is titled, Understanding Dyslexia, a Brain-Based Learning Difference. In his keynote, Dr. Odegard will break down the brain-based language differences of dyslexia and outline the risk indicators and characteristics of dyslexia and how these can be targeted as part of an intervention grounded in structured literacy. Following the keynote, we have a variety of breakout sessions for you to choose from, including screening, instruction, and intervention for English learners within an MTS framework, fostering accessible instruction for all, intensifying foundational reading instruction to meet the needs of all learners in spelling, visible language to inform instruction and intervention. I hope you enjoy the PLN, and thank you so much for being here to learn with us to improve reading outcomes for all of our students.
Thank you, Superintendent Gordon. Good morning and welcome to the Spring Professional Learning Network. My name is Tammy Wilson and I'm the project lead for the California Dyslexia Initiative and a director at the Sacramento County Office of Education. I am excited to learn with you today from our amazing presenters. As Superintendent Gordon noted, this morning we'll hear a keynote from Dr. Tim Odegard. You'll notice that there's both a chat and a Q&A section on the platform during this keynote. Please share your questions and we'll get to them if time allows. Dr. Tim Odegard is a professor of psychology and holds the Catherine Davis Murphy Chair of Excellence in Dyslexic Studies at Middle Tennessee State University. He leads the research laboratory and efforts of the Tennessee Center for the Study and Treatment of Dyslexia. Dr. Odegard is a trained educator who has worked with students with reading disabilities and completed a 2-year dyslexia specialist training program at Texas Scottish Rite Hospital for Children in Dallas. He serves as an editor-in-chief of the Annals of Dyslexia and also served on the editorial board of the Perspectives on Language and Literacy. He was a member of the Understood Parent Advisory Committee and currently serves on the AIMS Scientific Advisory Board. Dr. Odegard received the Luke Waits Alta Award of Service in recognition of significant contributions to improving services for individuals with dyslexia and the IMSLIC Innovator Award as an outstanding MSLE professional. As a developmental cognitive psychologist, his research focuses on memory and language. His reading research focuses on 3 areas: identification of children who struggle with reading, intervention for students who struggle to read, and teacher training. I know you will get a lot out of this keynote. I have enjoyed learning from him over the last few months, and please join me in welcoming Dr. Tim Odegard.
Thank you so much for that warm welcome, and thank you to everybody who's here right now sharing your time with us. And I hope that you are safe and well in California, as I know from many of my friends, like Marianne Wolf, who I was speaking to in conversation with a few weeks ago, and my buddy Julie Washington, who I'm picking up from the airport later on today in Nashville, that you have been through a lot in California. You are with us And I hope that your family and you and your loved ones are safe in these times. So I just wanted to kick off with that. I'll be talking today and trying to keep a pace that's appropriate. I've got a slide deck. These will be available. And if I don't go through every single one of them, please don't fret. Hopefully you'll have a chance to see other aspects of this later on. My first keynote is going to be more high level, kind of a thought conversation to pure curiosity, if you will, in you as a listener. My breakout session will be much more nuts and bolts and go over some case studies and examples of screening and literacy profiles and risk profiles, moving into kind of a high level understanding of structured literacy and what we've learned about what we should do for instructional practices for all learners, but most importantly, those like myself and my son and my sister who have dyslexia. One sec. Hold on one second. Not technical difficulties, just got to get the proper screen there. I'm going to be covering 6 big concepts, which I'm going to link to a big idea. It's just an idea that I pulled out. It's not the big idea, it's one of many. I would encourage you at home to play along. Maybe as I'm presenting each of these different sections, you come up with what your big takeaway is. It's not the big idea that I'm going to share with you, it's just one of many. The first topic that I want to cover is a conceptual model of dyslexia, which I often get asked to speak about. Now, this is the abridged version. This is the one that I often give, and this is linked back to something I developed back in 2020 in a publication. The reviewers were actually pretty critical of this one. They thought it was too simple. And from a research standpoint, it is very simplified. But I do think from an educational standpoint, as educators, which I am, as you heard, and as maybe diagnosticians, which I help to train out of my center as we train PhD and master's students in literacy and literacy studies, as well as school psych students from our psychology department in more nuts and bolts of actual assessment for literacy profiles. I think it's an appropriate level for that, but we're going to dig a little bit deeper as I move past this. So let's look at the abridged contemporary model. It's broken into 3 main parts: the distal causes, which we have understood from dyslexia that give rise to a behavioral presentation of dyslexia, and those intervening proximal causes that are right in between those. The behavioral presentation is clearly defined. Those of us with dyslexia have inaccurate and slow and labored word reading. We have inaccurate, inefficient decoding. That's applying the alphabetic principle of, let's say, the English language, which is an alphabetic language, to read words accurately and efficiently with automaticity. We often also have spelling deficits. It's not just a reading disability, it's a written language disability. It impacts both our ability to read words accurately as well as spell words accurately. And we are really hard to teach. So we'll be in your classrooms and your other little kiddos are learning, but we're lagging behind. Maybe you put us in a little bit more and you try to intensify that intervention and maybe a tier 2 kind of a setting. We're still slowly progressing and definitely not doing what Joe Torgerson said to do, which is to close the gap to allow us to catch up to what we should be doing. Now, more proximal, these intermediate kind of things is it's often found that those of us with dyslexia have phonological processing deficits. Those are often going to be our awareness of the individual sound structures in words, those individual units, the phonemes, or maybe the onset sound and maybe the rhyme that follows it. So that in the at in cat. It's really hard for us to learn the sounds and those graphemes that we use to represent those letter and letter patterns that go along with them, such as trigraph TCH. And we lack automaticity. One of the last things that ever comes around, if it ever does for us, I'm still very labored and slow when I'm reading text that I'm not very familiar with its syntactic structure. And funny enough, technical papers like neuroscience or cognitive science, I read quite efficiently. But if you were to give me a novel and you asked me to read it, I would be so slow and labored, it would be painful for you to hear. And I'm a grown adult who is the editor-in-chief of a research journal. I have published over 60 articles in my life, and I have contributed to so many things with written language. Yet the ease with which I access that language and write that language is very difficult for me. We also have these issues with automatically accessing the sound structure of language, what Marianne Wolf calls rapid automatized naming, as well as others, in addition to Marianne, call it that. And sometimes that's wrapped into that phonological processing and accessing orthographic patterns, which would be those spelling patterns that I mentioned. Those distal causes, well, it has a genetic basis. There's brain structures involved, there's brain function involved, and how we connect those brain regions to functionally do tasks are also impaired, or not impaired, different is how I would like to say it. That's the abridged version. We cannot forget that all of this is impacted by our environment, our home environment, the schooling environment, and support structures around us, as well as that we come with both intrinsic things to us, which will protect us, as well as risk factors inherent to us, but also around us. We have protective and risk factors. But I want to go into the expanded version for you for a couple of reasons. One, when people who are actually trained neuroscientists like myself, who also are educators and can help to translate this in hopefully a way that gives you at least a foothold to summit this mountain of evidence that we've amassed, I'll fill a void that could be filled by misinformation, because other people who aren't neuroscientists might decide that they want to speak to these pieces to you, and they may not be as informed. And what I have learned over my life as an educator is, once I learned a lot, I learned how much more there was to learn. I learned what I didn't know. And as a neuroscientist and as a cognitive scientist, I know so much and I've contributed so much, but it's such a small fraction of what we know and what we have left to learn. I find that knowledge begets humility and an awareness of what we don't know. And so I would like to share with you a more comprehensive, expanded view, which is still just the tip of the iceberg. And I'll also be prefacing what I say with a lot of caution. But what I don't want you to ever take away is what we do know. And the most important thing is dyslexia has a genetic basis. And we've known this for a long time because of the twin studies that were done by people like Dick Olson at the Colorado Center for Reading, as well as ongoing research by research scientists such as Sarah Hart at the Florida Center for Reading Research, using twin studies to look at this. More recently, with the sequencing of our genome, we've been able to move into the frontier of molecular genetics and actually start to pull genetics apart into their basic components. We're in the beginning of this phase of looking at it for what it means for many behaviors that we present, not just dyslexia. So I want to unpack a little bit of this and what these different layers of this graphic that I've done mean. The first is, as you would know, because you probably have had a science class or two, our brains, our bodies are programmed by our genetics. The majority of our genetics is shared with almost every other species on this planet. It's a small part of it that allows us to differentiate into our species, and an even smaller part of that genetics that allows us to differentiate into our own unique butterflies that we all represent, with our own unique set of strengths and weaknesses and gifts for the world. So at the core of who we are is our genetic composition. Variations in a very small set of these genes give rise to the differences you see in our behaviors, how we present. The study of this is called behavioral genetics. Twin studies are one method of study used to explore behavioral genetics. Molecular biology and genetics are another, and there's different types of methods that have been used and are continuing to be used to explore. What I want you to know is, we know that dyslexia is a genetic-based learning difference, grounded in our language development first and foremost, and is what we call polygenetic, meaning that there's multiple pieces on that double helix that come together to increase the risk that will present with the behavioral phenotype or the behavior characteristics of dyslexia. I hope that I'm going slow enough. And also, again, this is really basic. I think this is just review for most of you. I'm not telling you anything that you don't know as far as genetics at its basic components. Because we've done molecular genetics now for some time, there have been lots of candidate genes put forth at a molecular level. And here are a bunch. This is just a small fraction of them. Here's the takeaway from this slide, though. There's two big takeaways. One, they highlight differences in how our brains develop, in the migration of neurons, as well as the structure of key areas of our brain. Why would the migration of the neurons be so critical? Well, in the 80s, there was a Geschwin-Galiberta hypothesis that kind of was the second version of what they were proposing, that they were doing with Gordon Sherman and Glenn Rosen, and critical groundbreaking research that was using autopsy studies of donated brains from those of us in the community with dyslexia. What they were noticing was that there seemed to be layers of cortical striation. Let me just pull my brain out here. And key areas of the left cortex right here that were overshooting. Here's the temporal lobe.
Dr.
Where they
Odegard,
yeah.
you have your camera off, so if you're pointing to your brain, we aren't seeing it.
I should start my video. Key areas right here. You've got your temporal lobe right here. I can pull it apart. And then right here's your parietal lobe. What Gordon, Al Galiberta, Norman Geschwin, and Glenn Rosen were seeing was it looked like the migration of the neurons in vitro, in the uterus, were overprojecting here. What we've subsequently learned from molecular genetics is a potential genetic basis for that, which is what you would presume. What's the second thing? Brain structure. How some of these areas are structured, such as the research of Nadine Gobb, who's highlighted key areas in the areas that process the letter patterns seem to be structurally different pre-reading instruction in children with dyslexia. What are we also learning? These genes are linked to oral language development and how we process language. So what are the two big takeaways from the molecular genetics? The candidate genes all point to oral language development early on and how we process the phonological structure, as well as how we access through repeated automatized naming, and we can name objects. And the migration of cells in our brain early, if not in the womb, as well as the structural development of key areas in pieces of the reading network, which I'll present in section 3, I believe. That's the key takeaway from this slide. You don't have to know what every single one of those genes are. Science is ongoing. We'll find more, and we'll show that some of those probably aren't actual genes that are impacted. What those genes give rise to, that intermediate level, is what we call endophenotypes. An endophenotype is this intermediate level between our genetic predispositions and the behavioral presentation, that behavioral phenotype. So it comes in between. Here are several candidate phenotypes. I've listed 6. 4 of these are mission critical that we have very strong evidence for. The last 2 have been proposed, but there's far less solid evidence for. I want you to see that the strength of evidence across research will point to certain ones that have been considered, while others. I want you to know that it isn't like we've just looked for like a handful of these. We've actually looked for quite a few of them, but some bubble to the top, such as phonological processing deficits. One of the most important phenotype is that we often find phonological processing deficits in those of us with dyslexia. An endophenotype will be more likely to be found in people who have genetic risk, the polygenetic risk, will be more likely to be found in the families of people with a history of dyslexia, and they're not deterministic, meaning if I have a phonological processing deficit, it doesn't mean that I necessarily will have the behavioral characteristics, but I'm in an increased likelihood. Also, not everybody who presents with the behavioral characteristic will have that endophenotype. And that's because of a lot of important things that can happen with the interactions of all the different levels on that graphic I showed you. Rapid automatized naming is another one. I was just reviewing most recent research, and there was a publication in 2023 from a set of Chinese researchers that were looking at a candidate gene, one of the ones that I had in that slide. They were showing that it was manifesting itself into a link with rapid automatized naming and brain regions associated with that, and a sex-linked genetic protection for females that if they presented with the gene would make them less likely to have the presentation of dyslexia, giving us a potential mechanism, a biological mechanism for why some people say that dyslexia is more common in boys than girls. which is still, I would say, somewhat of a working hypothesis because we're not resolved in the research community. Some people like Jack Fletcher and Reed Lyon, Lynn Fuchs and Marsha Barnes have reviewed the research and said, well, it might be an over-identification in boys opposed to girls. Or people like Rick Wagner and others will say that it is more common in boys. Genetic research is critical from a basic scientific standpoint for us to still invest in, because it allows us to understand and go deeper to find out if there's reason to believe that there is a sex-linked difference between those at the presentation of dyslexia, such as boys having a larger presentation than girls. Orthographic processing is another endophenotype that we see. We struggle to process the spelling patterns, and spelling is one of the hardest things to remediate when we look at the clinical data or the response to intervention data or intervention data like I published in my time and career. Also, my clinical data would also show that. I have continually found that the automaticity with which we read and read words is hardest to remediate, as well as the spelling patterns. And basic science is critical here. I've been working and others are working to try to find creative solutions to use other learning pathways to allow us to overcome these lingering challenges that are difficult to remediate in those of us with dyslexia. Neuroanatomical differences are another. So those genes, as I said, and I highlighted, are associated with key neuroanatomical differences in language centers in the brain, as well as visual processing centers that are supportive of reading. Visual attention. Now, you've got a scholar in California, Dr. Jason Yateman at Stanford, who's doing cutting-edge research in this and is one of the leading experts on this in the world. And when me and I talk about things like this, we know that we're still in the nexus of trying to understand this. There's also critical research happening in the Italian language with several different independent groups in Italy who have been publishing on this for some time. Visual attention potentially has a role to be played. We're not exactly sure what it is, nor how solid the evidence is. So it's kind of a promising endophenotype, but not one of those top four. The other one is kind of important for us to put a caveat on, working memory. And when I say working memory, you probably think of executive function, and you think of taking some kind of a domain general task. If working memory plays a role, it plays a role through language. Critical to think about this. If you are anybody who does testing on, let's say, testing PA, and if you've ever given the comprehensive test to phonological processing, the CTOP, you know that there's a lesion. And a lesion, by definition, is a phonological working memory task. You have to do a task like, say, tiger, but leave out the g. You're using working memory. You've activated your secondary auditory cortex in your left hemisphere here, and the storage of that phonological information and interfaced across the fasciculus here, this white matter tract, to coordinate and access and use the executive function pieces up here to act on that. So how we measure working memory and what measures we do are how language dependent they are. The ones that seem to be associated with dyslexia are the language dependent. More recently, we've had a neuroimaging study published just a few years ago in Cortex by Dr. Lori Cutting, a leading expert in the neurobiology of literacy and dyslexia at Vanderbilt University using brain imaging techniques. She's also one of the oldest brain imaging researchers looking at reading comprehension. And also out of University of Houston with Jack Fletcher, who published neuropsychological data highlighting the same thing. When we see a link to working memory being predictive or associated with literacy and dyslexia, these are language-specific measures, not domain general, not a digit span task per se. But think about a digit span task too. You're activating phonological memory when you do a digit span task. And if you think, and you've been trained in the Baddeley model from the 1970s, there's the phonological storage, there's the visual spatial storage, there's the central executive, the executive function, and the interaction of those. It's the phonological side of things, the language-based side of things that are predictive. So this endophenotype is not a working memory domain general. What the evidence says, if it is a phenotype, endophenotype, it's language specific. The behavioral phenotype is what we see. We call it behavioral genetics for a reason. We're finding associations and causal pathways from those central genetic components And then there are variations at that level up to see what leads to the higher likelihood of behaviors that we have as humans, which is super cool if you ask me. But it's all surrounded by the environment we live. I pulled these environmental factors from a couple of publications that I may get to the slide or not, from Hugh Katz and Yakov Pesher, a publication that they had in 2021, and then a publication from Haif, Myers, and Haft that they had a little bit earlier, maybe in 2016, maybe in 2018, maybe in 2019. I've got the reference there. And if you go to the page on your portal for this, you'll see a link to both of those references. And I have references as well as resources for every single thing that I'm saying. I don't have a reference section. I actually give you links to the evidence and the research that supports what I'm saying and practical tools that you can access from those. That behavioral presentation for dyslexia is inaccurate word reading, spelling, inefficient, slow and labored word reading, slow response to generally effective instruction. That's our behavioral phenotype. That is what dyslexia is. Those are the primary characteristics. Environment impacts those. If we have a strong home environment, like my son had, where I gave him language intervention on the spoken form from day one, my son, unlike me and my sister, never presented with phonological processing deficits that were detected by the CTOP, because I intervened on his phonological processing. But what could he not do? He couldn't read words and couldn't spell words and was slow and labored, and the school instruction wasn't addressing his needs. So we got him a structured literacy intervention for 2 academic years at a high dosage, which is what my research and others has shown to be highly effective. But his behavioral phenotype didn't quite look like it should have. With his behavioral phenotype was there, but an endophenotype was missing. So let's pop up to the endophenotype. My son did not present with a phonological processing phenotype, unlike myself, who couldn't actually hear syllables in multisyllabic words when I was in an AP English class my junior year of high school. Nor could my sister. She was in an AP English, but she couldn't either. So an endophenotype may not materialize. It isn't necessary condition to identify a condition. This is the foundational basic science that undergirds the recommendations you hear from people like Jack Fletcher, Famiko Haif, Rick Wagner, and others when we talk to this. And one of the big things that Rick is really pushing for right now is to move away from a deterministic all or none causal mechanism to more of a probabilistic likelihood. My son shouldn't be denied an identification of dyslexia because he never presented with a phonological processing deficit. It was clear that environmental factors from birth had intervened and helped to mitigate the manifestation of what his genes probably made it more likely that he would have. And your behavioral phenotype interacts with this. Once you start reading words and learning how to read words, you develop a phonemic awareness. Before we actually have to start looking at the print, we never need to get to individual phonemes. We just play nursery rhymes. So I need to be able to go cat and hat and bat and sat and mat and fat. I'm not at the phonemic level there, except for the onset, you would say, but I'm not into the medial or the final position. I've isolated the onset and the rhyme. But once you start doing synthetic phonics, you are actually engaging in a phonemic level of awareness that's at the phoneme level. And that's because you're actually intervening there. So as you develop the ability to access language, you will develop a higher proficiency with your phonological processing. But if you can't access, and it's hard to learn and to understand that, and you can't access words, then it's going to exacerbate your phonological processing. They go hand in glove together. Genetic variations are right here. I've listed just the 6 candidate genes that I put there. I gave you a more recent review on this from a stunning researcher out of Texas A&M from 2022 or 2023 that you can read if you want to, and you can look at what she has to say about this, as well as the fact that there is some caution needed. Where the caution isn't needed is the substantial evidence that there are genetic basis from all the family history studies that have been done and the twin studies that have been done for decades now. and the environment will influence the genetic expression. Anybody who's had to take one of my infancy and early childhood development classes knows that teratogens are environmental toxins that will directly impact the expressions of your genes. There's just one example of an environmental factor that can impact your genetic variation. But the core of us as humans are our genetic underpinnings that gives rise to all of the layers above nested within the environment that we grow up in. So what's a big idea? And you can write down your own here. The link between oral language and dyslexia is encoded in our DNA. The link between oral language and written language is encoded in our DNA as a species that has evolved for so long. So let's talk about language and where it comes from. This is an evolutionary biologist here, Pagel, from one of the universities in England. I just love this reference from him. Possessing language, humans have had a high fidelity code for transmitting detailed information down the generations. Many, if not most of the things we make use of in our everyday lives rely on specialized knowledge or skills to produce. The information behind these was historically coded in verbal instructions, and with the advent of writing, it could be stored and become increasingly complex. Why do I like this? It honors oral language. And in our country, we have proud communities, especially our indigenous communities, who have a proud current oral history tradition. Language is something that we have had as a species for a very long time and is part of who we are and part of our culture and our cultural integrity and honor, and we should honor that. 2, we invented writing. It's a human invention. And it's all language. Now, I give you the link when you go to the portal of the ASHA documentation that actually is far more complex than this. This is an oversimplification. What I want to drive home is reading is language. Writing is language. It's written language. It's all language in its receptive and expressive forms. You are a literacy teacher. You are a written language teacher. You are an oral language teacher. You are a diagnostician who is looking at language, written language and oral language. Never forget your roots. And there's some key things that I want you to know and remember about this. 1, oral language came first. 2, reading must be taught and practiced. And don't forget the practice. 3, we never lose oral language, except for those of us who are otherly abled and we're born or acquire a difference that means that we co-opt and develop neurodivergence that would allow us to use language processing centers for the oral components, such as in the temporal lobe and the secondary auditory cortex. They're otherly abled and they will develop capacities for those if they are born or acquire a hearing difference. So I do want to honor that neurodivergence there. We have spoken language to communicate for a very long time. And the specific number of years is a ballpark figure here, but a whole long time. As a result of this, there are very specific integrated regions of our brain which will develop because they are coded in our DNA. And that genetic expression will give rise to our capacity to have integrated systems in our brain that allow us to acquire naturally our mother tongue and other languages in the oral traditions that we are exposed to and are taught. Reading is a newer human invention. And again, it goes back a few different ways. If you've heard my podcast, Dyslexia Uncovered, you know that I talk about the fact that there's 3 nexuses. There's the Saharan African birth of written language. There's the Mesoamerican birth of language, and there's the Asian Chinese birth of language. Writing was developed, some say independently, across different aspects of the world. So no one culture can claim the right to say that they invented writing. It was coming about in different systems. One of the cool things that you can say is specific to those different traditions is the Chinese culture gave rise to what we would call a logographic syllabary or syllable level way of capturing our spoken language and writing. And our history back to the Greeks, the Phoenicians, and the Mesopotamians and others will give rise to an alphabetic language where we use individual letters, groups of letters we call graphemes, to represent single phonemes. So there is no cultural dominance when it comes to language or written language. And I think that's key for us to keep and have cultural humility when we think about these things. Children learn to understand and produce oral language from exposure. Speaking to babies culturally around the world, we often call motherese, where we parse out. Through pragmatics, we engage in joint directive and attention to teach them objects as well as verbs. Children must be directly taught how to read and write in all languages, all written languages. And for what we call in alphabetic languages, more shallow orthography, such as Spanish, which has a more of a one-to-one consistent correspondence between the letters and the sounds they represent, that direct instruction leads to a quicker pace of learning to be accurate with that. And dyslexia will have a slightly different shape and shade of its presentation in shallow orthographies relative to a deep orthography like English. It takes practice to become automatic readers and writers in every writing system. And how we structure that practice and how we scaffold that practice as teachers is critical to the success of our learners. And because of those genetic underpinnings, as well as their manifestation into those endophenotypes, there will be individual differences. Folks like me, my sister, and my son are much harder to teach and much slower to respond. Some of us having lifelong struggles with key aspects. I just tweeted something else out yesterday, and I had misspelled a word in it because I didn't have all my assistive technology wrapped around me with Grammarly, and I was doing it after I got a medical procedure in my cervical spine, but I wanted to get it out, and I misspelled things. But that's okay. I understand who I am, and I understand that I have some limitations, and I understand that through AI and assistive technology, I can have scaffolds that support me, but I feel no more shame anymore, and I've released the shame. Oral language to continue to develop and expand even after we enter school and formal education. And this development interacts with reading, especially that word study piece. As we start to read, we can read more complex word formations and patterns as far as how we put words together. The syntactic structure gets more complex than our spoken language use, and that's critical. We are experiencing far more words and more elevated words and more academic language, depending upon what we're being exposed to in written language that we can acquire, which is mission critical and opens up opportunities for us to pursue various pathways to express ourselves across the span of our lifetimes. And to give a shout out to my buddy Marianne Wolf, who I cite at the bottom of this, engaging meaningfully and deeply with text fosters vocabulary, background knowledge, creativity, and innovation. It's critical that we honor oral language as well as written language. Also critical to understand that no one culture or society has a monopoly on the invention of written language. And we can represent and have a cortical view. This is the outside of this brain. If you actually pop open and you look inside of a brain, you'll actually know that there are subcortical pieces here. So if you pop it apart, you can actually see that there's subcortical regions like the basal ganglia going down to your brainstem here. Those aren't the cortical. So when we say a cortical model of reading, we're talking about the convolutions and layers on the outside most of our brain, something that we have as humans. Dolphins are really good about this too, and packing a whole lot of brain mass into a small nut or the shell that is our skull. Let me put the pieces of my brain to the side here. All these different parts will be part of a network of regions that allow us to engage in certain behavior. This is a graphic that comes from a book that was published by Stanislav Dehaene. We all struggle with pronouncing his name. He's French. Have some grace here. Anyway, I love his graphic for a few reasons. One, it's color-coded for networks of nodes that interact with one another functionally to bring about a human behavior, such as the orange areas that allow us to pronounce and articulate spoken words and also process the speech sounds of spoken human language. Also, those green areas, which are critical for the processing of the meaning structure of language. And then you'll notice that red area, which is very important for processing the orthographic patterns, the letter patterns. We have yellow, which is attention, and blue. Now, what I want to do is I want to build for you the reading brain. In the center, you see a representation of actual human behavior performance in fMRI scanners around the world from over 14,000 studies that we have synthesized into that brain for you. These people were lying in the scanner and reading or looking at reading patterns, word-written strings to make this activation. So you'll see all those purple areas representing what I just showed you on Stanislav's cartoon or picture, the drawing of the brain. I'm going to build the reading brain, and I'm not going to actually present any print to build it, because reading builds off of oral language and then puts on top of that other key ability. When we looked at those 14,000 studies and we said, let's look at places where they're hearing spoken language, you see that top brain up there and it's yellow. You'll notice that that maps to areas of when you're in the scanner and you're reading. Then what about when you're processing meaning? Across those 14,000 studies that we pulled out, those that were processing meaning, you see those green areas. Notice how those map onto the reading brain. How about across those 14,000 studies about the studies that were involved in attention? Notice how attentional areas that we add on to get reading map on to the reading brain. And notice how the visual processing centers in the brain, when we asked those 14,000 studies, we don't want the studies that had any reading in them. We want the visual processing that wasn't reading. Map on. Reading is the compilation of our oral language capacity to extract meaning, coupled with attention and visual processing in our brains. And that's pretty groovy, because I can build you the reading brain with never ever once showing a single lick of print to a child. But if you show a child print time and time again, you will co-activate all these areas which are involved in speech and hearing, processing meaning, attention, and visual processing, which is all part of the component processes that allow us as humans to read. And it's not just English. So there's an English reading brain. The left side is your left hemisphere. The right side is your right hemisphere. Here's Hebrew, another alphabetic language, which is also deep. I'm not going to go there. I always don't ever say too much, that I can actually just back up real quick. Spanish, which I know is a shallow orthography, right there. And there's a logographic. There's Chinese right there. Notice at how similar the cortical regions are that are involved in actually interacting with all these different writing systems from different areas of the world. Big idea here. Written language builds on oral language. It's language. All day long, it's language. Language is key. From birth, language is key. I'm looking at the time. We got 15 minutes left, so I'll keep going and get you the primary characteristics of dyslexia, because I'm going to let this part out of my breakout, knowing that everybody would hopefully be seeing the keynote. Now, many states around the country have adopted the 2002 consensus definition. That has been adopted by the International Dyslexia Association and was also adopted by the National Institutes of Child Health and Human Development here in the United States. And there's key parts here. One, it's a very dense, wordy definition. So it talks about its neurobiological origins. I hope that you have an appreciation of the complexity of the neurobiology, and also that we're not just pointing to one or two studies done in a few labs here in the United States. That there's genetic work happening in Germany, in Northern Europe, in China, in Australia, in the United States, in Canada, all over the globe. That there's brain imaging research that I just showed you images from those 14,000 studies from all over the world. I just showed you images of brain imaging studies from a synthesis of studies from different language orthographies. And that dyslexia has been characterized in more than just English. And it's not just because we have a squirrely language that we have a problem for people like me, my son, and my sister with learning how to read and spell. Now, as educators, that neurobiological origin is good to know as grounding, to know that there's something that we build dyslexia off of. But we're not going to do brain imaging scans to know how to work with our kids. And we're not going to do genetic marker tests to know if they have dyslexia or not. So it was actually a little superfluous and not necessarily needed in the definition, but it's there. The primary characteristics, that behavioral phenotype, is really critical. You have to have that in the definition. And we're really bad at reading and spelling words. And we're really slow and labored when we read words. Now, that endophenotype that we have the strongest evidence for, which we don't have to find because, as I said, it's probabilistic, not deterministic, is phonological components of language being deficient in those of us with dyslexia. And while not necessary and mission critical, because we struggle to read words, it can have downstream consequences in so many areas. So some of you may have seen this before. Young-Suk Kim is one of your professors at one of your institutions. She has a much different reading house that's built off of all of her tremendous research that she's done for a long time. This is my very simplified reading house. I called it the reading wall, but then we kind of wanted to build a wall and it got political. So I just call it the reading house. I don't like to put politics into any of this. So it's a reading house. Sorry, Suk Kim, I didn't mean to steal your metaphor, but I think we did this around the same time. And it's kind of like the simple view of reading the way I built it. If you know the simple view, it says that language comprehension and word recognition come together multiplicatively to give us reading comprehension. But I call the less simple view of reading because we know that especially language aspects of executive function are critical. But I'm going to take that away because we're going to focus on the language side of things. We know that, well, one, phonological awareness would be on the left side, but it's so critical for the print, it's going to pop up on the word recognition. But vocabulary development, oral, our understanding of morphemes, initially, those affixes, especially the suffixes, we learned very early. There's a very long research documenting the development of those. How we put words together to make utterances and sentences, and then other things are really critical, syntactic structure, as well as background knowledge. Those undergird are some of the pieces that undergird that give rise to our ability to comprehend written language. On the word recognition side, we have letter knowledge, their names, their shapes, their formations, and the sounds that they are paired with. Phonological awareness at the, let's say, onset and rhyme, as well as the phonemic level. Decoding and encoding, that using that sound symbol relationship to sound out and pronounce words and also spell words. And then word recognition. Everything in the English language is not going to follow a sound symbol correspondence. Most do, but not all will. So then I often highlight that there's a need for us to also address sight words and develop a sight word ability to see those. The main focus of your reading instruction should never be teaching sight words, but because of the English language, it becomes kind of a mission critical piece to be able to do that. The, for example, you're not going to run into a passage that doesn't have the, but in running text, that's how you pronounce it. And unless you wanted to say that it's in an unaccented syllable, but it's not because it's a single symbol word, it should be the, but it's often said the in running text. Accurate. We have to be accurate. We have to be automatic. Automaticity is facilitated by rapid naming, but we don't teach rapid naming for its own self, like naming objects or letters, for example, because we can practice rapid naming by just reading and naming strings of letter really quickly that we call words or sentences or paragraphs. So if you want to practice, do that structured practice to give them opportunities to transfer what they're learning in basic word study to actually read real, authentic, connected text with your support and supervision, going back to the word as your way of scaffolding, and then reading fluency. And all of that supports reading comprehension. So there is one simple view, less simple view of reading. Those are the primary characteristics of dyslexia. We're really bad at decoding and encoding. We're really bad at word recognition, and we're slow and labored. Often associated with that endophenotype. And I split these two up. So that would be the phonological processes and the PA. And then I split it up into the rapid naming. So there seems to be different gene loci that are associated with different variations of those. And the secondary consequences that are named in that 2002 definition. Because you're not accessing printed language, you're not developing your academic language and vocabulary or being exposed to new words that you might not hear in your daily conversations. And you're not being exposed to build up your background knowledge. So knowledge building is critical for reading comprehension, as you're well aware as educators. Now, in October of this year will be a special issue of the Annals of Dyslexia on the definition, that 2002 definition. And I've invited, as the editor, a whole host of stellar experts on dyslexia. Hugh Katz and the entire crew at the Florida Center of Reading Research are one of those contributors. They highlight that if we were going to talk about secondary consequences, we should talk about the very real mental health challenges that those of us with dyslexia experience. Researchers reported that almost 60% of those children with dyslexia meet the criteria for at least one mental health issue. And right now, it's pretty clear that the internalizing issues are the ones that are most prevalent. So many studies have documented high proportions of anxiety and depression among children with dyslexia. And there's just a few citations down there that go to support what I just said. There's currently, and there's a link to this in the portal, the most current issue of my journal that I edit, the Annals of Dyslexia, has a special issue on these mental health conditions. If you want a collection of recent studies and meta-analysis on this topic. And they too come to the conclusion that most of the research points to internalizing. However, the externalizing behaviors can manifest as well, but there's far less research on those. What's the big idea here? Or one of the big ideas that I kind of take away from it, and you take away your own. Dyslexia is characterized by inaccurate and inefficient word reading and spelling. And there's a whole host of things. I've got another couple of sections that I didn't get to, but I'm also not going to try to run through and skip slides and highlight this. But quickly, I can do this because this is like 2 slides. We have strong evidence that there's associations between structural brain differences, functional brain differences, differences in brain connectivity, the pathways, the white matter that connects the brains, as well as brain chemistry, the neurotransmitters themselves that are signaling and allowing our neurons to work in the brain. Most of this research has been done in 2 fronts, the structural differences using structural imaging, as well as functional differences using functional imaging. I was the second person that ever published a paper on the connectivity using DTI after Bruce McCandless, who's a scholar and a professor at Stanford there in California, had published his first paper on it. There's 3 main areas that we see differences in. Those areas that allow us to process the sound structure of spoken human language. And then what I like to say, this hub area that puts together the letters and the sounds to bring them together, which is up here in these areas. If you can see, I don't know if you can see my cursor, but hopefully you can. If you can't, it's no big deal. It's those top 2 blacked out circles up there, those nodes in Stanislav's figure that I've represented here on this picture of a brain. Actually, the one that I was pointing at earlier, it's a picture I took of it. The brain's letterbox is that red area. And Nadine Gobb has shown that in preschool kids with a family history of dyslexia, you already see structural brain differences in those areas. And we also see functional differences with less activation in there in those of us and children and adults with dyslexia that are not remediated yet. And areas in the frontal lobe show different types. And I say different because some people show increased activation there, some show decreased activation. We show aberrations relative to a neurotypical or your normative kind of what we would say is the normative development. Our neurodivergence will come in different shades up there in those frontal lobes. There are clear evidence of what we need to have. Here's a recent meta-analysis from Colby Hall and her crew at University of Virginia. And you'll notice that we should directly teach phonological awareness, word reading and spelling, reading comprehension, and give lots of practice with text reading. Dosage matters from their meta-analysis. The more is the better when it's direct instruction with lots of practice, as well as these domains are the ones that they found to be significant predictors of reading outcomes. And my research and others shows that we can turn on key areas of the brain in response. Those areas that do the speech pound production, as well as in the inferior parietal lobe that do the integration of speech and print, will come online and show a normalization of activation relative to typically developing readers. And the letters, the brain's letterbox there that processes orthographic patterns will also show increased activation. Exploring brain-based differences with dyslexia highlights the role of language, because how much of that I highlighted was language, orthographic processing, speech-human sounds, and mapping those together. And I'm going to stop right now because you need to get onto your breakout sessions, and I'm not going to take any more of your time. And I hope that you found value in sharing your time with me, because you could have been changing the lives of your children and your students. Thank you for this time.
Thank you, Dr. Odegard. That was, wow, that's a lot to think about. We have 3 minutes left and I have like 30 seconds of closing. So in that 3 minutes, there is one question that I'm wondering if you can speak to. Someone asked, they've attended different trainings on dyslexia and noticed that there's been an uptick in conversation around morphological processing and its effect on reading. And is this a component of dyslexia?
So yes, I would say it is because of its relationship of morphology influencing spelling patterns and pronunciation. We have a morphologically complex language in our alphabetic writing in English. You can read the writings of Louisa Moats on this, as well as Marcia Henry are foundational. One of the things that happens is us researchers take this very slow and labored way of thinking about it. And the word nerds spent all of the 80s and early 90s looking at single syllable words and simple affixes that are a closed class, your suffixes, S, ed, ing, es, er. There aren't that many. Derivational morphology, where we bring in our Latin and Greek forms, which actually are really important to know. Is I don't know if you've got Elsa presenting later on. Elsa's going to be traveling here later tonight. I think she's actually in Houston, and I think she may be doing one of your breakouts, and she has to pit stop, get a hotel room so she can present to you guys, and then hop on a plane to get to Nashville. But she left earlier to get there to Houston to get into a plane from Brownsville. So she's doing a lot of convolutions to get there. But when we talk about it, those derivational Latin and Greek forms are really important in building on our multisyllabic words. And when you do that, you'll often have spelling shifts that happen, as well as pronunciation shifts. Think about shun. You can spell shun different ways. It's always pronounced shun. It always has the same meaning. Think about it, something as simple as adding a vowel suffix to a closed syllable base word that causes a doubling. When I look at finite data with our dyslexic population, something as simple as a doubling of skip to skipping, and you double that P, we see a steady decrease in their ability to accurately read that on microgranular level reading measures that we've been looking at for some time now. So yes, I would say that morphology is important, but it's important because of how it's situated in the structure of the English language. And morphology is critical for pronunciation, for spelling, and for meaning. It is really important glue that binds all of those together in our morphologically complex language. And where you see it the most is in our multisyllabic words. And the word nerds didn't get around to multisyllabic words until recently, so they didn't really highlight the importance of morphology until recently, if you want to look at the history of this in the research. However, our practitioners, like Louisa and Marcia Henry, they've been talking and thinking about this for a very long time. So is Anita Archer. She has as well. So sometimes research has to catch up to what we've already been seeing and knowing we had to address from a clinical standpoint, from an instructional standpoint. I know we're all educators on this, and I don't want to use those clinical, more medical terms. We're educators, and our educational leaders who have helped us, who have instructional methods, have been knowing about this for some time. So I think that's not a simple answer, but I think it's an honest answer to the question. But it's not true for just dyslexics. It's true for lots of readers. And you'll notice as educators, and probably have, that reading small, simple words that are very consistent is one thing for many readers, but you'll have a lot of readers. It's what Don Compton studied and then Hollis Scarborough studied, and they called the late emerging form of reading problems. They didn't show early stage problems, but when they get to the big chunky words, they struggle.
Well, thank you. All right. Well, you have given us a lot to think about. The second question in the chat was around the PDF of Dr. Odegard's slides, and it has been re-uploaded. So hopefully the new version doesn't have the sides cut off. Well, I hope all of you learned as much from this presentation as I did. It definitely left me with a lot to think about, and I'm going to want to revisit the presentation. So remember that the recordings will be available on our platform by next week or maybe sooner, and they'll be available through mid-April. So if you need to revisit that because your brain is exploding right now, it will be available. Also, Dr. Odegard is hosting a breakout in our first session. So if you want to learn more, you can join that session. On another note, we want to take this time to invite you to our CDI conference, which will be June 11th and 12th. And actually, Dr. Elsa Cardenas-Hagen is one of our keynotes, and Dr. Odegard just mentioned her. So Wynn will put the research or the link and the flyer in the chat. We have a fabulous lineup, and we hope you can join us. We now want to invite you to join a breakout session. If you've already built a schedule, it will appear on your Event Mobi homepage. And if you did not select one ahead of time, just go to the sessions on the left and go ahead and choose one. You will enter the Zoom by selecting the Join Now button, and it will take you directly into the breakout room. Please, we do encourage you to fill out the feedback survey because we pay attention to that and it helps us with planning for our upcoming events. And then there will be a second breakout session. You will enter the same way. And after the second breakout session, we encourage you to join me back here for a closing. And I hope you will all enjoy your morning, and we will see you soon. Thank you so much for listening to the keynote, and I hope you enjoy the breakouts.